Okay, so how then do we get rid of the neurotransmitter? I can't just leave the neurotransmitter there. If I do that, my cell will keep trying to fire and keep trying to fire and keep trying to fire. That's not going to work. So how do I get rid of it? One of them you already know. The first one, enzymatic degradation, you already know that one because you saw it in chapter 9. We talked about the acetylcholine esterase and how it takes acetylcholine and it breaks it apart, basically making it so that it doesn't work anymore. So that's one way we can do that. We can also do take backsies. It can be taken back into the presynaptic terminal. Some neurotransmitters are really expensive to make. They cost a lot of energy for the cell. I don't just want to break it once it's done. It's not like, I go out and I buy a $100,000 car and I drive it once and then I take a sledgehammer to it. No, you keep using it. Why? It was a $100,000 car, right? So with some neurotransmitters, okay, like what we see down here, again, horrible color, think I'd learn, down here, norepinephrine is a good example of this. Norepinephrine is one of the neurotransmitters we have, and it's expensive. So I release it and it goes across and it does its job, but then when it's done, I pump it back and I just reuse it. Why? Because it's expensive. So I'm basically recycling. That's what I'm doing. Now, the other thing, the other way we can get rid of it, you have to remember that the neurotransmitter only works on the cell that it's next to. It only works across this space here. That's literally the only place that this functions. So if I let it float away from that space, it's not going to do anything else anywhere else. There's nobody who has the right lock for that specific key. And sometimes neurotransmitters are cheap. If I've got a cheap neurotransmitter, I can let it do its thing and then it just floats away, eventually working its way into the urine and you pee it out. But those neurotransmitters have to be pretty cheap. Even when it comes to the enzymatic degradation that we were talking about with acetylcholine, we still saved part of that. If you'll remember, we broke it up into acetic acid and choline, but we took choline back and then we put acetic acid with it. So it's not like we totally got rid of everything. Enzymatic degradation means we're going to break it down somehow. If it's broken, it doesn't work, so we basically get it out of the way. Taken up into the presynaptic terminal, we release it, we bring it right back. Okay, we reuse, we recycle. Diffusing out of the synaptic cleft. In other words, I release it and it goes over here and then I just let it go out into the body. And that way, it's not in that space, so it's not going to hurt anything or cause another action potential. Specificity and neuromodulators. Okay. Neurotransmitters are specific for their receptors and they can either be inhibitory or excitatory depending on the receptor that they bind to. Okay, first thing in this sentence that I want to talk about. Specificity. I can give you a key to my house and I can tell you, go through Alice and find the door this key belongs to. Will you ever find the lock that belongs to that key in Alice? No, because I don't live in Alice. I live in Kingsville. So if the key doesn't fit the lock, it doesn't work. I have to have the specific key for the specific lock for this to work. And neurotransmitters are specific for the things that they bind to. In other words, if I've got acetylcholine and it's a norepinephrine receptor, that isn't going to work. That's like me giving you a car key and telling you to go open somebody's front door. It's not going to work. It's not the right key. It doesn't fit. Now, we kind of mentioned excitatory or inhibitory before. There are times when I want things to speed up, and there are times when I want things to slow down. The easiest way I can think to explain this is fight or flight. We talked about the sympathetic nervous system and that it was fight or flight. And if there's a, cougar in my backyard, excuse me, if there's a cougar in my backyard or a mountain lion in my backyard, my heart's going to speed up, right? The neurotransmitter that goes to my heart when there is a mountain lion in my backyard is norepinephrine, also known as adrenaline. Adrenaline gets dumped into my body, and my heart goes because my body's going, you better get ready to fight that mountain lion, or you better get ready to run, one or the other. Either way, we need the heart beating faster so that the blood's moving faster. Yes? Okay. That is an excitatory response. That specific neurotransmitter is designed to get my heart to speed up. More action potentials, faster heart rate. Let's talk inhibitory. If I'm in rest and digest watching The Masked Singer on TV, right? Am I going to be, oh yeah, I have to make sure that, my heart's speeding and everything? Why? I mean, The Masked Singer is exciting and all, but it's not that exciting. I don't need my heart going a mile a minute for that. So when I'm in rest and digest like that, what do? Well, I slow down my heart. I release acetylcholine when I'm in rest and digest, and that goes to my heart and slows down the action potentials. It basically calms it down. Why? Because if I'm watching TV, my heart shouldn't be beating like I'm in a Zumba class. That's why. Okay? So, inhibitory, I'm going to prevent the action potential. Excitatory, I'm going to cause the action potential. Now, something else. Some neurons release more than one neurotransmitter. It's true. We have neurons that release multiple neurotransmitters. Why? We got nothing. We don't know why it happens. We know it happens, but we have no idea why it happens. So we have to kind of take that into account sometimes when we're talking about this stuff. Now, for us, we don't really have to worry about it because it's not like I'm going to specifically say, okay, guys, so, no. But I do want you to understand that it does happen. Neuromodulators. To modulate means to control. Okay? So there are substances that are released from some neurons that can either pre or post-synaptically influence the likelihood that an action potential in the presynaptic cell will generate an action potential in the postsynaptic cell. So we have chemicals that actually get released that either increase the likelihood that your next cell is going to have an action potential, or they decrease the likelihood that you're going to have an action potential. Either way, we call it a neuromodulator because it's controlling that neuron. Is it going to have an action potential or is it not? I'm deciding because I'm the neuromodulator on that chemical. By the way, a neuron is a single cell. A nerve is not. A nerve is a whole bunch of cells all braided together to make something bigger. Okay, excitatory postsynaptic potential versus inhibitory postsynaptic potential. Postsynaptic, the cell after the synapse, after the space. Excitatory neurons. Excitatory neurons release neurotransmitter that will cause the postsynaptic cell to depolarize. Just the word excitatory should tell you what it's going to do. It's going to cause the cell to depolarize. When depolarization occurs, the response is stimulatory, and a graded potential is called an excitatory postsynaptic potential. It's usually the result of an increase in sodium. Well, that makes sense. Sodium is really high outside. If I open those channels, sodium should rush inside and make the inside of my cell more positive. That graph with my depolarization, this is becoming more positive. So that makes sense. Inhibitory postsynaptic potential. The name gives it away. Inhibitory, okay? You do have inhibitory neurons that release neurotransmitter that will basically cause the postsynaptic cell to hyperpolarize. Now remember, here's my resting membrane potential. If I want it to fire, it goes up like this. But if I make it hyperpolarize, it's going to go down further than resting membrane potential. It's going to get even more negative inside the cell. Now, the best way that I can think to explain this to you, by doing that in the cell, it makes it even harder for it to get positive inside the cell. If I were to say, let's say we're in the classroom at the school, right? And I were to say, go to Burger King and get me a hamburger. You'd probably go, oh yeah, it's not that far. Okay, sure. And you could walk to Burger King, right? Now let's say I drive you to Houston and I go get me a burger from Burger King in Alice. I'm pretty sure you'd go, are you crazy? That distance increased, making it a lot harder than just walking across the parking lot to get to Burger King, right? So when we do this, when we hyperpolarize this cell and we make it even more negative than resting membrane potential, it makes it harder to get to an action potential. So inhibitory neurons release neurotransmitter that hyperpolarizes that postsynaptic cell. When that happens, the response is inhibitory. I'm going to basically reduce my chances of having an action potential. And we usually call it an inhibitory postsynaptic potential, okay? It usually results from an increase in the permeability of potassium, where all of a sudden I'm leaking potassium out of my cell, making it more negative, or an increase in permeability of chloride. Remember, chloride is negatively charged. It'll flood into my cell, but it's going to increase the amount of negative inside the cell because it is itself negative. Presynaptic inhibition or facilitation. Inhibition, to inhibit, facilitate, to help, okay? In this example, in the picture, I have something called an axonic synapse. There's an axon, synapse to an axon. Axonic synapse. Ta-da! Okay? Don't let the word freak you out. It's just an axon touching another axon. So, an axon of one synapse is connected to the axon of another synapse and can release a neuromodulator. Remember, neuromodulator means to control, right? Now, that neuromodulator can influence how this first synapse works. So, I've got presynaptic inhibition or presynaptic facilitation. To inhibit means to stop. To facilitate means to help, okay? So if you look at the example here on the PowerPoint slide, here is one axon terminal, but it's inhibitory. If you'll notice in this first picture on the left, this is not releasing anything, right? Look at how many neurotransmitters are getting released by that first neuron. A lot, right? Now, look over here. Now this is actually releasing neurotransmitter. Look at this one. Did I stop it completely? No. But is it as effective as it once was? If you look from left to right, you'll notice that this working actually inhibited this neuron up here from working very well. So it's presynaptic inhibition. I'm inhibiting it. If it went the other way, where without it, it was releasing very, very little, but with that presynaptic axon releasing its chemical, all of a sudden it's working like gangbusters, that would be presynaptic facilitation. It would be helping it to release its neurotransmitter. I was trying to erase the actual figure. Yeah. Spatial and temporal summation. Spatial. Think of actual space. How big is the space in front of you? How big is the space behind you? Actual area. Okay? Temporal. Think time. Okay? If you've ever heard of a temporal rift in any sci-fi movie ever, it's a rift or a rip in time. Okay? So we talked about trigger zone already. We talked about that when we were looking at our neuron. We had our neuron, it had its axon with its axon terminals, and that trigger zone was right after the axon's cell body. It had the axon hillock, that cone-shaped portion, and then the initial segment of the axon. The reason we call it the trigger zone is because of this. It has 7 times more of the voltage-gated sodium channels than the rest of my neuron, meaning that it is highly sensitive. A little bit of sodium going through this area goes a long way to getting an action potential. That's why we call it the trigger. Okay? So, spatial summation. So when neurotransmitters are released by several synaptic bulbs at once, so it's across a larger area. If you look at this first figure here on the next slide, you'll notice I've got axon terminals here and here. I've got them on 2 spaces along my axon. Spatial summation. Action potentials 1 and 2 cause the production of a graded potential at 2 different dendrites in 2 different places. These graded potentials summate, they sum up, at the trigger zone to produce a graded potential that exceeds threshold. What happens if I reach threshold? I fire. I get an action potential. Now, temporal summation. Neurotransmitters are released by basically over time. So I'm not releasing neurotransmitters from different places. Now I've got the same axon terminals, but I'm releasing neurotransmitter, releasing neurotransmitter, releasing neurotransmitter, releasing. So it's over time. I keep doing it over and over. So neurotransmitters being released several times by the same synaptic bulb. So again, if you look at this picture, I've only got one set of axon terminals here. But if I release neurotransmitter, wait, release neurotransmitter, wait, release neurotransmitter, wait, release neurotransmitter, eventually this is going to build up and I'm going to get an action potential in this neuron. Now, EPSP, excitatory postsynaptic potential, in combination with IPSP, inhibitory postsynaptic potential. It's a competition, and whichever has the most active axon terminals wins. So, what do I mean? Okay. Hang on. There. If you look here at this picture, down here, I want you to focus down here, okay? Don't worry about these now. I've got axon terminals that are excitatory, 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 excitatory. But I also have some that are inhibitory. Who has the higher number? I have 3 excitatory versus 2 inhibitory. Basically, an action potential is produced at the trigger zone when the graded potential is produced as a result of EPSPs and IPSPs summate to reach threshold. Pretty much the larger number wins. I have 3 excitatory neurons firing and I have 2 inhibitory. Who has the higher number? The excitatory. So the excitatory is going to be the one that in this case will cause an action potential. So, circuit. A circuit is a specific pattern. If you've ever done circuit training at the gym, you repeat the same pattern. I go to the leg machine. I go to the treadmill and walk for 2 minutes. I go to the arm machine. I go to the elliptical and walk for 2 minutes. I go to the leg machine, right? You repeat the pattern. So I've got 3 different types of circuits that I'm going to talk about. I want you to know, though, there is a 4th circuit that you're going to have to know that is going to be in your homework. So you have access to the ebook. Use it for your homework assignment. Diverging circuit. To diverge means to split off. Nerve impulse from a single presynaptic neuron causes stimulation in increasing numbers of neurons. So I start with one neuron and it basically excites 2 neurons, which excite 2 more neurons. So I'm getting bigger. I'm adding to my signal. Okay? Converging circuit. I've got several presynaptic neurons and they're actually combining basically to get to this one neuron at the end. Okay? Oscillating circuit. If you think about an oscillating fan, it repeats the same pattern over and over, right? And it goes to the left and then comes back to the right, and it goes to the left and comes back to the right. It repeats that pattern over and over. In an oscillating circuit, you have collateral branches of the postsynaptic neurons synapsing with the presynaptic neurons. So you've got these neurons coming down, but then it feeds back to another neuron. So that pattern repeats over and over and over again.